Okay, this should look familiar because we did this in class. If you don't have yours filled out, then you can use these notes to fill them out now really quickly. So the way this works was interphase up here. We already went through that in the other two videos, so not going to talk about it now. So we're going to start right here. Number 2 would be prophase. Okay, so in prophase, we have our nucleolus and our nuclear membrane start to disappear, so that's right there. We use dashed lines to show that they're breaking down. Your chromatin condenses into chromosomes. Remember we talked about those as being sister chromatids. Our microtubules, our spindle fibers are forming right up there. They kind of look like little spider leg things. I always thought they did anyway. And those form from our centrioles, okay, which are those little cylinder things. Remember we said their job is to help in cell division. We said that a couple chapters ago when we were talking about organelles. So that's prophase. So nucleolus and nuclear membrane disappear. Your chromatin is going to condense, and your microtubules or spindle fibers start to form. All right, then we move down to number 3, and this is going to be metaphase. So we're going to put an M. The way I always remember metaphase is meta starts with an M, and what are your chromosomes doing? They are lining up in the middle of the cell. Your spindle fibers are going to attach to the centromeres. That's what those dark spots are supposed to be in the middle. Those are supposed to be our centromeres. Remember, our centromeres are going to hold together those chromatids in the sister chromatid. So the centromeres hold together the chromatids, and the two chromatids hooked together are going to make our sister chromatid or our chromosome. So metaphase, they meet in the middle. All right, so now we have anaphase over here, number 4, and I always remembered anaphase starts with an A, and what is happening are sister chromatids are being pulled apart. So what happens is those spindle fibers shorten up, and when they're shortening up and contracting, they break the centromere, and that allows those chromatids to separate. So now what you end up with is, you can see we have 2 distinct sides here. Important to recognize this at this point, because what are we going to have at the end? 2 new cells. So what's going to happen is both these sets of DNA are being pulled to the opposite side of the cell. I'm sorry, not the cell, of the of what would be the cell, and then we're going to make the new nucleus. Sorry. All right, and then the last part of mitosis, because remember we only have 4 phases of mitosis. We have prophase, metaphase, anaphase, and now we have telophase. And again, I remember this because tele starts with a T, and 2 starts with a T. That looked like an I. So we have 2 new cells starting to form. So what's going to happen is our chromatids, remember, got pulled apart, and so now our nucleolus can start to come back. Our nuclear membrane is going to start to come back, and we can see that with the dashed lines, and we should have 2, and we do, because we're going to start ending up 2 new cells. And then our cytoplasm is going to start dividing. Remember, it starts, so you can start to see it pinching in right here. Okay, and then at the very, very last, the second part of the M phase, okay, separate from mitosis, is going to be cytokinesis. Okay, what does it do? It divides cytoplasm, and we're going to get 2 new identical cells. Why? Because we have the same copy of DNA in there. We can see our nuclear membrane is back. We have the 2 cells. One thing I want to point out that would be different is if this were a plant cell, okay, instead of getting this like pinched off area over here, what we would end up start to form is that cell plate. If you remember, we talked about that before. So there's our chromosomes and DNA. And what's going to happen is we're going to get this cell plate that starts to form across the 2 cells, and that's what's going to separate your cytoplasm. The purpose of this whole thing, why we have mitosis? Well, in some organisms, in single-cell organisms, it can be used for asexual reproduction, right? You're just making a copy of itself. That's what asexual reproduction is. No sex needed. And then in multicellular organisms like us, what we're going to end up doing is replacing old cells with it, or we can use it to help us grow. Okay, remember we said embryos and children are going to go through mitosis quicker. Why? They're growing. Okay, so they're going to have a lot of cells that need to stack up on themselves so they can get larger and larger and larger. Okay, if you have any questions, see me in class. And again, if you didn't get your notes filled out in class, you can use this to help you fill them out now.